At the end of the learning session, the student must be able to understand concisely the function of enzymes, explain properly the models of enzymatic reactions, enumerate completely the different classifications of enzymes, and lastly, know correctly the different uses of enzymes. The word enzyme came from the Greek words enzyme, en meaning in, enzyme meaning yeast. An enzyme is an organic compound that acts as a catalyst for a biochemical reaction. They speed up multiple cellular reactions as compared to those uncatalyzed reactions. Take note that during these catalytic reactions, enzymes are not being consumed, but instead, they just merely help with the reaction to occur more rapidly. Most enzymes are globular proteins. Some are just simple proteins consisting entirely of amino acid chains. Others are conjugated proteins containing additional chemical components. Until the 1980s, it was thought that all enzymes were protein. But nowadays, a few enzymes are classified as ribonucleic acids or RNA and catalyzes cellular reactions involving nucleic acids. Take note that if a certain chemical is needed in the cell, the enzyme responsible for its production is activated by other cellular components. When a sufficient quantity has been produced, the enzyme is then deactivated. There are two main types of enzyme structure: a simple enzyme and a conjugated enzyme. A simple enzyme is composed only of proteins or amino acid chains, while a conjugated enzyme has a non-protein and additional protein part. The protein part is known as the apoenzyme, while the non-protein part is known as the cofactor. The holoenzyme is the biochemically active conjugated enzyme produced from the apoenzyme and a cofactor. Take note that by itself, neither the protein nor the non-protein portion of a conjugated enzyme has a catalytic property. Cofactors provide additional chemically reactive functional groups beside those present in the amino acid side chains of apoenzymes. A cofactor is generally either a small organic molecule or an inorganic ion. They are usually a metal ion. The typical inorganic ion cofactors include the following. We have zinc, magnesium, manganese, and iron. There are various vitamins that have coenzyme functions in the human body. Coenzymes are small organic molecules that serves as a cofactor in a conjugated enzyme. So how do we identify if a certain substance or molecule is an enzyme? Enzymes are most commonly named by using a system that attempts to provide information about their function rather than the structure. Their type of reaction catalyzed and substrate identity are the focal points for this nomenclature system. A substrate refers to the reactant in an enzyme catalyzed reaction. It is where the enzymes act upon too. There are three important aspects in naming process of enzymes. Most enzymes ends in the suffix ase or ase. For example, we have the enzymes urease, sucrase, and lipase. But just like for most of the rules, we have an exemption. The digestive enzymes trypsin, chymotrypsin, and pepsin ends in the suffix in, which is found in the names of some of the first enzymes studied. Another aspect is the type of reaction catalyzed by an enzyme and is often noted with the prefix. For example, we have oxidase, an enzyme that catalyzes an oxidation reaction; hydrolase, an enzyme catalyzing hydrolysis reaction. The last aspect includes the identity of substrate and the type of reaction catalyzed. Examples are glucose oxidase, pyruvate carboxylase, and succinate dehydrogenase. Infrequently, the substrate but not the reaction type is given. as in the names of the enzymes urease and lactase. So for this type of enzymes, the reaction like hydrolysis is involved. Let's have a review of what we have discussed so far. Enzymes speeds up biochemical reactions. Most enzymes are globular proteins. There are two types of enzyme structure, the simple and conjugated enzyme. The substrate is the substance upon which the enzyme acts. Most of the enzymes ends with the suffix ase, but the first studied enzymes end with the suffix in.